Welcome, everyone. My name is Nick Cisneros. I'm a commercial marketing manager here at Oxford Nanopore Technologies. I apologize for the technical difficulties here. I think we got all that resolved, so we're good to go here. We're going to get started. I'm delighted to welcome you to this webinar where we will be hearing from Philipp Rescheneder and Anthony Doran discussing scalable human genomic characterization with nanopore sequencing. After the talks today, there will be a live Q&A session, so please add your questions to the Q&A box, which you'll find at the bottom of your screen. You can also upvote the questions that are currently there or that other people put in that you would most like answered, and we will try to get through as many as we can. This webinar is being recorded and will be made available on demand after the broadcast. So it gives me great pleasure to welcome the first presentation today from Philipp Rescheneder, who's our Senior Director, Applications of Bioinformatics at Oxford Nanopore Technologies. Philipp and his team benchmark and adapt the most appropriate tools and workflows for analyzing Oxford Nanopore data in the context of a wide range of applications. Thank you, Philipp.
Hi everyone, my name is Philip. I'm a bioinformatician here at Oxford Nanopore in the applications team. And today I want to talk to you about characterizing human genomes at scale with nanopore sequencing. So what's the plan for today? Here at Nanopore, lately we have had a strong focus on providing end-to-end workflows, basically from sample to answer. And today I just want to give you an overview of three of those, focusing around human genome sequencing. Mainly I'll be looking at what I put here as human variation, which would be germline, whole genome, human genome sequencing. So use cases like rare disease or population genomics. But then in the end, I just want to give you a brief glimpse at what Nanopore can do in terms of cancer genomics and single cell transcriptomics as well. Right, but before we dive right in, let me just give you one brief slide of introduction, saying what do we mean when we talk about characterizing a human genome. So generally what it comes down to, if you sequence a human genome, what you want to find, what you want to detect is obviously genetic and epigenetic variation. And there's a huge range of different genetic types of genetic variation. And to frame it a bit, I typically use a quote from a paper from 2016. And so it says kind of like the main thing that it's about is kind of uncovering the genetic basis of human disease and phenotype. And if you want to do that, what you need to do is you need to understand the pattern of human genetic variation. That's the first part. And as I said, there's a huge variety of different ones, small SNPs and indels, as large SVs and CNVs, and then also things like epigenetic modifications, for example. But that's only half the picture, because the quote goes on and says you don't only need that, but you also need to understand the haplotype structure of the human genome. You need to resolve which haplotype these variants are on. And so this is basically, in a nutshell, what I want to show you today using nanopore sequencing. So let's start at the very beginning with small SNPs and indels. So as you probably know, you can Nanopore will allow you to detect SNPs at really high precision and recall. So what we did here is we used the typical genome and bottle data sets to benchmark the performance, and found a really high performance at 99.7 F1 and above. This, what I show you here, is a very simple example. The important thing is that it scales to regions that have historically been harder to map. So here I have an example for you. It's a repetitive region that's hard to map with shorter reads. So there's a lot of reads that don't have very high mapping quality on the bottom, but on the top with long reads, you see all the reads have high mapping quality and the variant is very easy to see. And this also extends to small indels, like this frameshift deletion that I'm showing you here, a typical example from the BRCA gene, especially in coding regions. We can call these at very high precision and recall as well. And most importantly, this analysis is all wrapped up in our end-to-end workflow and our workflow human variation. But again, this is just kind of like the first part of that quote that I just showed you before. This is just about detecting the variation. We then also talked about phasing those, or resolving the haplotype structure. So how does that work? So basically, the important thing that you have to do when you want to phase or resolve the haplotype structure, you need reads that span multiple variants. And so in this kind of like cartoon here, if you see at the top, if your reads are much shorter than the average distance between your variants in your genome, then that won't work. You'll have one variant per read or no variants at all, and then you really can't phase. But as your reads gets long and longer and exceed the average distance between variants in your genome, you'll get reads with two variants or three, and then you can kind of start piecing them together. You see this one variant in one read and in the other read, so you know they come from the same haplotype. But then these have two more, three more variants, and you can do the same. And so you can, again, piece it together and puzzle together and kind of tease apart the reads into two groups. And one group would be from haplotype 1 and one group from haplotype 2. So you can not only call the variants, but also phase them. So to illustrate why this is important, I'll give you a real-time example, like an IGV screenshot from it. This is kind of like a region of the CFTR gene. You can see we did exactly what I just said. We teased the reads apart into two groups. The top one, the pinkish one, is haplotype 1, and the bottom one, the blue one, is haplotype 2. And so these colored bars, you see our heterozygous variants. And so here what's annotated here is there are two variants that are known to be pathogenic. They are splice variants. And so the important thing here, if you don't know which haplotype those two variants are on, you don't know whether one haplotype has both. So if only one copy of the gene is affected, or if both haplotypes are affected, meaning both copies of the genes are not functional anymore. And then if you can phase, as you can see here, it becomes clear as day. You can easily see that one of the variants is in haplotype 1, the other one is in haplotype 2, so you know that both haplotypes are affected in this example. So with this, so far we've been talking about small variants, right, SNPs and indels. But as I said, kind of like a huge part of genetic variation in humans is part of larger structural rearrangements. And one thing about these structural rearrangements is that they often hide, so to say, in repetitive regions or in segmental duplications. There's one example here where there is a 4 kb segmental duplication around the HBA1 and 2 genes, so the alpha globulin genes. And what you can see here is the point is, if your reads are much shorter than those 4 kb, it's really hard to tell whether a read comes from one copy or from the other, so it gets really hard to map to. But if your read length exceeds those 4 kb, it becomes trivial. As you can see here, the reads map with high quality across that region, but you can easily see that the deletion that was called overlaps with HBA2 on one haplotype, because these are even phased, again, on the top haplotype 1, on the bottom haplotype 2, so that even extends to SVs. Again, this is of course only one example. We did human bottle benchmarking, and we find high precision, and we call typically independent of type and size of the SV, and kind of like the numbers that we get. It now is mostly limited by how good the truth set is. And again, this is all wrapped up in our human variation pipeline. There are many other really interesting kind of parts of the human genome that have historically been hard to call. And one more that I want to show you is clinical repeat expansions. So on the left here, I just have one example, the FMR1 gene. And again, the reads are phased. On top, you have haplotype 1. On bottom, you have haplotype 1, 2. And so on top, it's to see there, although there is a small insertion, it's really the repeat isn't expanded. It's still in a very healthy range in terms of length of the repeat expansion. Whereas on the bottom, you have an expansion of the repeat that's multiple kb long. But nevertheless, although it's multiple kb long, it's easily spanned by nanopore reads. And so you can easily characterize this. And that's also part of the human variation workflow. But this is actually not where the story ends for this example, because one of the factors here is that it's not only about the genetic changes, but also the epigenetic changes. And since Nanopore is native sequencing, you not only get genetic information, but also epigenetic variation. So basically, with the click of one button, you can switch around the coloring and you can visualize 5MC or 5HNC in these samples. And so what you would expect in this region is that one haplotype is kind of epigenetically silenced through imprinting, and the other one is unmethylated, so can be expressed. And in this example, we would expect actually haplotype 1 to be epigenetically silenced. And that's the case. The red indicates that it's modified, and also the track at the top shows you that there's basically 100% methylation frequency across the CpG in these regions. However, we would expect the bottom one to be unmethylated and expressed. However, with that large expansion, what we see is that the methylation frequencies are also very, very high, close to 100 here. So another important information for repeat expansion diseases. So this brings me just to modification calling in general. Again, the modification calling comes on top of like everything else that nanopore sequencing has to offer. So with long reads on the left, you can see you get access to centromeric and telomeric regions, and so you can even in those regions profile modification changes. Then in the middle, as I've also just shown you before, you can phase the read, so you get haplotype specific methylation. So you can easily find imprinted promoter regions. And then kind of most importantly, this really works at the click of a button, where you basically just toggle it on in our device software in Minnow. So far, I've shown you all these things in separation, but the real power comes that with a single data set, you can all integrate this into one. And this is one example that kind of like nicely shows it. What you have here is that, again, reads are phased. On the top, you have a haplotype with a large 8 kb deletion, so a structure variation that removes several exons of the mCOL1 gene. And then on the bottom, you have a haplotype without a deletion, but it has several heterozygous SNPs, and one of them is known to be pathogenic. And on top of all this genetic information, on the top you get the epigenetic information as well, so the 5MC frequencies. And again, our focus today is on end-to-end protocols. So basically for you to get to exactly this, we have an end-to-end protocol, and you can do that with a single PromethION flow cell for one human genome. And the analysis is kind of like automated on a Epitome platform. And so in the next few slides, I want to focus more on how you would actually reproduce this, how you could get from a data set, from a sequence data set to something like this. And again, for the wet lab side of things, for the extraction and the library prep and the sequencing, we obviously have step-by-step protocols. But then as soon as you have the data, we offer our Epitome platform that handles the secondary analysis of this. And so just to briefly introduce what Epitome is, it's basically two separate things. One, first, is a set of workflows provided by us. These are Nextflow workflows, and they're intended for certain applications to wrap up all the analysis that you would need. So for example, I've mentioned the human variation workflow for a couple of times, and I've shown you the kind of variance analysis you can get out of this. So this is one example. We also have a somatic variation, tumor normal pipeline, and for example, a single cell. These are just the ones that I'll touch briefly in this presentation on. There are many others that are available, and they're open source. You can run them by command line and kind of like run them on your cluster or any other compute that you would want. But the point is, you don't have to run it in a command line. As I said, this is one part of Epitome. The other part is the platform itself, which basically gives you a UI that can load all these workflows and will give you a user interface where you can input your sample name, your files, whatever analysis or parameters you want to specify. And then you can launch the workflow through that UI. And at the end of it, you'll get HTML reports and the output files. And this is just briefly shown here on the side. At the moment, this works mostly on local computers, but basically complete processing in the cloud is going to come soon and is in beta testing at the moment. So just to give you kind of like the end-to-end overview, obviously you would start by extracting your DNA, library prep, sequencing. The sequencing would give you base call data from the box. For this workflow, we recommend high accuracy base calling, so the devices keep up, for example, the P24. So at the end of the run, you get the base called BAM file with the bases and the modification calls. You could convert it into a FASTQ if you want, but then you would lose the modification calls. So you take that base called BAM, you put it into secondary analysis into the Epitome platform, either through the app or through the command line, whatever you prefer. And these will give you all your variant files, or read files, BAM files, phase BAM files, bigwig files, all these kind of things. And if you take this and put it into something like RGB, you can directly get to a screenshot like I showed you here. But I told you that some of these variants are known to be pathogenic. So how would we do this? And for this, we have another feature that's going to be introduced soon, which is kind of direct integration with third-party tertiary analysis providers, where you can directly load your VCF or your variant files into these platforms and use these platforms to then identify pathogenic, likely pathogenic, or otherwise interesting variants to give you kind of truly end-to-end experience for this workflow. So this brings me to the end of my my short overview of kind of like human variation workflow, basically the alignment-based human variation workflow. One quick glimpse I want to give you is that more and more people are switching over from an alignment-based approach to an assembly-based approach, where instead of just taking the read and aligning them to a reference genome, they assemble them first, so they create a haplotype resolved assembly. And Nanopore as a platform is uniquely placed here as the only platform that can provide you with true T2T alignments from a single platform. And what I'm showing you here is an example of human T2T assemblies. Here it's still highlighted where you would use 40x of high-precision duplex data plus 40x or 20x or 30x of ultra-long data. But just recently, we introduced improvements to base calling accuracy and showed that T2T assemblies are now possible even without the duplex data, just with ultra-long and 4C. And we're also working on integrating, for example, a read correction step into the RADA that's actually available right now, so that then kind of like can take the corrected data directly into state-of-the-art assemblers, and so this will allow you to reproduce what I'm showing you here. So there's definitely way more to come on this topic, and this will also scale to other organisms outside of humans. But as I said, today we're focusing on human genomics here. And so for the last 5 minutes or so, I just want to give you, as I said, a brief glimpse into cancer genomics and single cell. So let's start by cancer genomics. Cancer, in a sense, obviously is a very complex disease. So the hallmark, there are many hallmarks of cancer, for example, genomic instability, and there's many changes a cell has to undergo before it turns into a cancer cell line. So basically, complete view of all different types of genetic and epigenetic variation is incredibly important in cancer. And to come back to the point of uncovering the genetic basis of human disease and phenotype, I just want to give you a quick overview of what you can get out of a Nanopore run when it comes to cancer genomics. So we'll just start by looking at a human lung cancer cell line here. And what we did first is we just basically broadly screened the data sets for large copy number variation. And what we can see here is something very kind of like obvious, a massive amplification on chromosome 7. And so this is something that obviously has been possible for a long time with many different technologies. But since we have long reads, we can actually zoom in, we can like look at these sharp rises, for example, and see what happened here. So how was this region amplified so massively? So we can zoom in and we can look at the read alignments. And so, for example, here at the bottom, I show you kind of like 2 inverted duplication calls. They were again called with an SV caller. And these are kind of like known mechanisms of how these amplifications could happen. There are many more breakpoints in this data set, but this is just an example to show you can zoom in and you can understand how this amplification happened, and then step by step try to basically reconstruct what happened. But that's not everything. Kind of like the next question, like look at this obviously is kind of like why is this region amplified? Can we find anything that would give us a hint of why such an amplification would be beneficial to this cancer type or this cancer cell line? And so what we did is we screened this and have like amplified regions for SVs and smaller SNPs and indels. And what we found was this 15 base pair deletion in one of the exons of EGFR. And this variant is actually a known activating variant. So in a sense, very kind of like, simply put, it's beneficial for the cancer. And then again, here we can also, same as what I've shown you before, phase the reads. So we can actually differentiate between the two haplotypes. And what we can see here is that the top haplotype, which has this activating variant, has been highly amplified. Whereas interestingly, the bottom haplotype, which doesn't have this variant, it's just at its normal copy number, so there's no amplification at all. So this is just kind of like a brief example of like how you can kind of like piece together the puzzle of what's going on in the cancer data set. But this is again only the genetic side of things. Where it truly becomes very, very interesting is if we switch over to the epigenetics. And this is what I want to show you on the next slide. So what we did here is we sequenced 12 tumor normal pairs from 4 different tissues, 3 pairs for each tissue. And then we called methylation, and we basically tried to find interesting regions. And what we did was we simply said, please kind of like to our like differential method region detection software, DSS, please give us all the regions that have a consistent modification pattern within one cancer type, but that pattern is different from all the other samples. So it's a very broad and open question. And what popped out as one of the first examples was this region here. And as you can see, it's visually quite striking. There in this region, there's in most of the samples, there's no methylation whatsoever. And that's expected because it's a promoter region. However, in the 3 colorectal tumor samples, there's high methylation there. So that was quite interesting. I didn't know the gene, I'm not a cancer researcher, so I just went and Googled that gene, and what it turned out is that this is actually kind of like a known driver, a more important gene in colorectal cancer. And again, what you can see here is that the signal is actually quite clear. And that's interesting because we then also looked back, for example, at copy number vision, calling for these cancer types, and at least for 2 of them, there wasn't really a clear signal. And so that again just shows you how beneficial it can be to have both sides of the coin, basically genetic and epigenetic variation in one go. So this again was just a brief glimpse into cancer. There is much, much more that you can do. Please have a look at our tumor normal pipeline to see what else is in there, what else you can do. I'll just use the last few minutes to move on to single cell transcriptomics. Again, just a very brief glimpse, and I want to start by showing you why you would use nanopore sequencing for single-cell transcriptomics. So this is just kind of like an example region that's just the annotated and isoform from a gene called ACTB. And then this is a visualization of what you would get from short-read single-cell experiment. So you basically sequence kind of like starting from the end, so your reads will only kind of like extend kind of like a very tiny amount into the actual transcripts. So this allows you to get gene level expression, but not isoform level expression. And this is how it would look with a long read platform, where you with Nanopore you start kind of like you start at the end, but the reads are long enough to reach through your full transcripts. You get full length reads. And then with this, you can move to isoform level expression in single cell. And to do that, again, we have an end-to-end workflow that's also supported by an epitomy workflow that will do most of the secondary analysis for you. And just to, again, give you a brief glimpse in how it looks, I'm going to use just one example that we did in-house, where we sequenced memory B cells. And so what you often see with single cell is something like this. So this is from a short read run. If you do single cell, kind of like one of the first things people will often show you are these UMAPs. Basically, clustering of cells based on their expression levels, their gene expression levels. If you do the same with Nanopore, it would look very, very similar. However, again, you can move to isoform expression levels, and this actually does change the clusters quite a bit. And then the pipeline itself is compatible with analysis and exploration toolkits like Seurat. So you basically, with the output from that, you can start asking questions. And one question that we asked is kind of give us the clusters where we see the biggest isoform switches, or the most obvious isoform switches. And one thing that came out were these 2 clusters, and there is these two isoforms from BCL2, and you see they're actually quite similar. There is just a small difference in the UTR, but since we sequence the whole thing, we can even distinguish between those. And what we can see is that in cluster one, where kind of like one of the isoforms has 30% of the expression of the abundance, and the other one has 70 in cluster 5, this switches completely around to 10 to 90. So it's a clear isoform switch. That's something that you will just simply not see with a short read experiment. So again, this was just kind of like a very brief glimpse. I hope I've been able to give you an overview of our end-to-end workflows for these three different topics. There is much, much more, especially on the bacterial side and many other topics. But again, this was focused on human. I only briefly touched on the Epitome platform because my colleague Anthony will give you a much deeper dive on the analysis side of things. And with this, thank you very much for your attention.
Thank you, Philip, for that fantastic presentation. I would now like to welcome our second presentation from Anthony Doran. He's our Associate Director, Bioinformatics for Field Applications. Anthony will be presenting his talk on bioinformatics from beginner to production, Epitome, Oxford Nanopore's data analysis platform. Thank you, Anthony.
Hi, everyone, and welcome. My name is Anthony Doran, and I'm the Associate Director for Bioinformatics Field Applications in the Europe region. In this talk today, I'm going to talk about our bioinformatics platform, Epitome, and talk about how you, as a beginner in bioinformatics, can use our workflows through the Epitome platform, or as a bioinformatician, use these workflows in production environments to scale your analyses in an automated, reproducible, and robust way. So this year, one of the themes that you will have come across a lot among Nanopore data and our presentations is really this idea of helping you understand what you're missing really matters. And in this talk, I'm not really going to talk about applications where we can highlight new discoveries that are only accessible using nanopore data, but rather I want to talk about some of the performances that you can achieve from the pipelines that I'm going to recommend. So whether that's looking at, for example, industry standard, gold standard structural variation detection or SNV detection, or even if you're interested in resolving haplotype specific differences using phasing information. And again, this is all achievable from single sequencing run using nanopore data. Importantly, this also includes modification calling. So as I say, in this talk, really what I want to do is I want to highlight some of the workflows that we're producing that can help you explore your data to resolve these types of questions and also to get and achieve these types of performances and accuracies. But when it comes to bioinformatics software and the bioinformatics pipeline, I'm going to position this in the context of going from your sequencing right through to your analysis. So really what I mean is I'm going to talk about primarily the Epitome platform and how that links to our sequencing software, Minnow. And that's just really important to understand that in this context, you only really need to know about two software tools. There's Minnow, the software that sits on all of our sequencing devices and is used to manage the sequencing process. And then there's also Epitome, our analysis platform. And importantly, Epitome has both local options and cloud options, but also it comes with a point and click user face and also a command line implementation. So when we talk about Epitome, what we're talking about are tools and workflows that we produce that are available to help you understand both the quality of the data that you're generating, generate QC reports and assessment to ensure that the data you have going into a particular analysis is correct. But also these are workflows dedicated to analyze your data within a specific application or within a specific framework. And I'm going to show you a little bit of those workflows or show you what some of those workflows that we have available are in a couple of slides. What's also really nice about all of our workflows in Epitome is we also produce a report, and this report contains a summary of the sequencing run and the analysis, including those QC metrics and also plots and tables related to the underlying analysis itself. Importantly, Epitome can be installed on all of the main operating systems. So that includes both Windows, Mac, and Linux. It can also be installed on devices like a Promethean or a Gridiron, for example, to help you do analysis on device. So at the end of the presentation today, I'm actually going to show you a little bit of a slide with a summary of the compute resources required for various workflows that we also produce. But, as I say, what I want to get across to you is that we provide this platform that is designed to make your life easier, really, in a sense. What we want to do is we want to take the parts of an analysis pipeline that you would repeat across multiple samples and give it to you in an automated workflow that you can then deploy on your system. And by doing so, not only are you using the best practice recommendations that we would suggest, but you're also using a platform that's completely supported, user-friendly, and is making use of our best practices and will be updated alongside those best practices. What's also really important is Epidemy right now is completely open access. So you can download Epidemy right now from our GitHub page and from its website. You don't need to have generated Nanopore data. All of these workflows come with a demo data set already included, and also on the Epidemy platform or the Epidemy page, we have links to our open datasets portal. So if you want to replicate some of the benchmarks that we have published previously, you can install Epidemy, install one of the workflows listed here, for example, such as the human variation workflow, and run this on publicly available datasets like Genome in a Bottle. And again, what we want to do is we want to basically facilitate analysis in your environment. So like I mentioned, Epidemy comes in two kind of immediate flavors. There's a user-friendly interface or a point-and-click interface, and there's also a command line implementation. So this means that, you can start running it in your production environments in a very automated way, or you can even train wet lab scientists, or even wet lab scientists can use this through the point-and-click interface to perform the analysis themselves, especially if, time is critical and you really want to turn around analysis of a particular sample. Again, this is kind of minimizing the amount of time you need to spend processing or handling data, because these workflows are designed to basically take the output from Minnow and immediately go into our workflows. And that's essentially, what we want to do is we want to simplify that overall process for your bioinformatics pipelines. And all of our pipelines in Epidemy will take the outputs from our sequencing software, Minnow. So there's absolutely no need for intermediate processing. There's no need for intermediate filtering or any kind of additional handling of that data. It is literally a two-step process where you take data that's been sequenced and base called on our device, and that goes immediately as input into Epidemy, and Epidemy then performs the analysis. And some of the workflows in Epidemy, and this is really interesting because there are applications that will benefit from this, but they can be run in real time. And that means that as data is getting generated, you can start processing that data and you can start generating results. And in particular, we have workflows around metagenomics where this is quite often used. But this can also be used for on-demand analysis. So if you've completed sequencing, completed base calling, you want to analyze data, again, these workflows can obviously be used to analyze any data that sits on a hard drive it can access. But like I said, what we want to do is we want to facilitate the deployment of Epidemy in our workflows within a production environment. So we've produced command line implementations of these workflows, and that's really important to understand that the workflows that are available through the point and click interface are exactly the same as the workflows that we would use through the command line. And actually what the point and click interface is doing is it's acting as an interface to access, to download and run the exact same command line workflows that are available through our GitHub page. And that means that the parameters are the same, the expectations are the same, and the results are reproducible across both the command line and also the point and click user interface. But again, importantly, this allows you to then use it on a computer like a Windows, Mac, or Linux machine, but through command line interface or command line implementations allows you to run this on servers, computing clusters, or even in your own cloud infrastructure. And again, like I said, what we want to do is we want to essentially make your life easier by giving you workflows that we know are appropriate and best practice recommendations for various applications, but importantly, that can also integrate into your infrastructure. And again, these tools or these workflows, what we're recommending are not completely, wild or crazy. These are using industry standard best practice tools for various applications that allow you to then deploy these analysis on your infrastructure. And again, what we want to do, and I'll highlight this with some of the workflows that I want to speak about in the next couple of slides, is to generate results that are industry standard results. And what I mean by that is the output files that you would expect from a variant detection analysis will be the same files that you will have generated in the past with other sequencing experiments. And as an example of that, we would be looking to generate the variant call format or VCF format, which stores variants. And our workflows used for detecting variants will obviously generate this. In the same way, when we generate a de novo assembly, you'll get a FASTA formatted file, or when we generate other types of annotations, they will come in formats that are expected and as standard within that application. But again, I'm just going to highlight that in a couple of workflows. And as an example, I think one of my favorite workflows to always speak about is the human genome variation workflow, or WF human variation. And part of the reason why I want to talk about this is that this is a really nice workflow that can take immediately the output from our sequencing software, Minnow, and can do a lot of variant detection, annotation, assessment and summarization and aggregation of those results in a single workflow. And I'll show you what that looks like in the next slide. But essentially, through running this workflow, you can detect SNPs and indels or short variants, structural variants. You can do STR genotyping, CNV detection. You can do methylation aggregation, but you can also generate phasing information and start to integrate all of this information into a single set of analyses. And we generate a nice report at the end of this analysis that summarizes the variants that are detected across each of these variant classes, but also provides additional information like the alignment rates and the alignment profiles. So again, this is really nice example of a kind of simplified version of what we can do in a production environment using a single workflow, in this case, WF human variation, to analyze a lot of different variant classes. And what's really, really important is that all of these variant classes can be detected either independently or together within the same workflow. So for example, I might choose to run only SNV and CNV detection, and I can do that. Or I might choose to only do SV and methylation detection alongside phasing. And again, I can do that within the workflow. So I can select to analyze the particular types of variants that I'm interested in. And again, for anyone that's interested in, understanding, for example, the genetic etiology of particular diseases, this is a single workflow that provides a huge amount of information, like haplotype specific information related to resolving phasing, but also looking then at the variants that are identified within particular phase blocks, or if you wanted to look at methylation information. Again, this is providing files that allow you to understand haplotype specific resolution. And again, that's really important for understanding disease, or for imprinting, or for understanding any of the kind of large structural events that might lead to a particular disease outcome or phenotype. But in essence, this is what the workflow would look like from a production environment, where we have essentially sequencing is occurring and base calling is being done in real time on our devices, for example, on a Promethean. So what you're getting during the sequencing process and base calling is an output BAM file, unaligned BAM in this case, or it might even be aligned, that contains all of the sequence information and all of the methylation information. And this can be automatically transferred to a computing infrastructure using our data offload, like scripts and our data offload guidance within the Minnow software. And automatically, then these outputs can be taken into the human variation workflow to perform the alignment and the analysis and generate all of those results and those variants that I was speaking about. But behind the scenes, what this is actually doing is it's taking a lot of different tools and it's running them in parallel to generate different types of variants or different types detect different types of variants or different types of events. So, for example, I can see here that, this is using Claire Tree to do SNP and indel detection, or it's using Sniffles or Spectre for SVs and CMVs, respectively, or STRs for Straggler and ModKit for methylation. And one thing I want to highlight here is that what we're using are, in many cases, third-party tools that have been developed by the community that represent our recommendations for best practices based on benchmarking. So, we're not really doing anything in a black box way or behind some kind of, curtain where we're obscuring what's happening. We really want you to get the best results and the most reproducible results within our workflows. And what we want to do is incorporate those tools and provide a nice platform for you to run these in an automated way with the parameters that we would recommend for most of these applications. But I just want to highlight kind of one more example, and then I'll quickly show you a couple of workflows that are already available. And really, what I want to do is show you again what looks like a really complex process and how we've managed to simplify this down into a single example. And a really nice example of that is, of course, single cell, because single cell involves a lot of individual sequential processes that include both, the strand orientation of and the identification of full length reads, because again, if you have nanopore data with transcriptomic information, you can capture full length transcripts in a single sequencing run. But this workflow also includes all of the steps to do, for example, alignment, generate a transcriptome, annotate that, to generate counts, for example, of the gene by cell counts, for example, to do UMI assignment and correction. And then again, what we want to do is we want to generate reports that include visuals and summaries of this data for you. So again, this is a single workflow that takes many of these individual steps, but can be run very simply by taking the output from Minnow and using it as input into WF human or WF single cell with a single command, where simply the input is either a FASTQ or a BAM file and a reference genome and annotation. Then you can just run the workflow and generate all of these industry standard output files for you. But just really quickly, I also kind of want to jump through without going into any detail, a couple of other workflows that might be of interest as well, in particular that can be used in different types of production environments. And again, I mentioned single cell right there, but we also have a bulk transcriptomics workflow that can be used to identify, for example, the counts assigned to particular genes as an estimation for gene expression can be used to identify fusion genes, but can also be used to identify isoforms and annotate novel isoforms against a reference. And by doing so, allows you to then look at isoform specific expression and not just to look at gene level expression, but to go down to transcript level expression, which again is helping us resolve the kind of underlying biology around different types of tissues or different types of diseases or kind of experimental conditions. Another workflow, again, that's kind of like really, really powerful because it does so much in a single workflow where it takes simply the output from Minnow and will do a de novo assembly and annotate that is our clone validation. So again, this can support barcoded data or multiple sample data and generate full-length plasmids in a single analysis run. That again can be really quite fast. Similarly, for anyone that's interested in automating the analysis of bacterial data, we have a workflow for doing that again, where you can discover variants, perform a de novo assembly, and look at consensus annotation. But this also includes workflows for doing antimicrobial resistance detection annotation as well. So as I mentioned, there are a lot of different workflows available. What's really important to keep in mind is that the only workflow really that needs a GPU is base calling. That's the same as on our devices where GPUs are already integrated. And for almost all of the other applications, these are going to be CPU intensive or CPU require CPUs. But you can see here that obviously, the smaller the genome, small kind of like pathogen genomes or microorganism genomes tend to have the lowest amount of compute intensity and can be run on standard laptops nowadays, all the way up to, these genomes that are the mammalian genome scale, or these analyses at the mammalian genome scale, like human variation, tend to be a bit more computationally intensive. But really, this is just more of a rough guide for you to have as an estimation. But all of these workflows on the documentation page have recommended hardware compute for running the analysis with the amount of coverage and data that we would generally recommend per sample. So, that just brings us to the end of the presentation, but obviously, this shouldn't be the end of your Nanopore journey. If you are interested in learning more, you can always, for example, follow any of these links. You can always go to the Epitome website, download data and start analyzing or start running our workflows, even if you haven't actually generated data yourself. We have publicly available data, or you can always reach out to us either through the community or through the support email if you have any questions or want to talk about your data or projects, or how we can support you deploy our workflows in a production environment. So with that, I'd just like to say thank you for your attention. I hope you enjoyed today's talk.
Thank you, Philip and Anthony, for your time and presentations today, and thank all of you out there for attending. To learn more about today's topic, you can find more information using the link or QR code that's up here on the screen. If you have further questions about your projects or getting started with nanopore sequencing, please contact support@nanoporetech.com. This webinar, along with all of our previous broadcasts, are available for you to watch on demand, so please visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinars and knowledge exchanges at the Oxford Nanopore website. We'd really appreciate your feedback on this webinar. You can find a short survey on today's broadcast in the Q&A panel at the bottom of your screen. And again, we apologize for the technical difficulties at the start of the broadcast today, but I think we resolved those rather quickly and efficiently. So this will remain open once the webinar ends for the poll tab on feedback. So please provide us some feedback. We'd love to hear from you guys. So thank you all and enjoy the rest of your day.